Today's topic is based off of cancer and cancer treatment, today's topic is based off of cancer treatment, which is often a multi-pronged approach. One of the more common treatments for cancer is through the CHOP method. CHOP is a four-drug treatment composed of cyclophosphamide, an alkylating agent which damages DNA. Hydroxyanorubicsine is an intercalating agent which damages DNA by inserting itself between DNA bases. Oncovin, which prevents cells from duplicating by binding to the protein tubulin, and preadenosine, which are corticosteroids. Over the course of the next 2 weeks, we will examine these drugs. And for today, the focus of the topic is the prevention of duplication by oncovin. As I said, today's topic is describe how oncovin binds to tubulin and leads to prevention of cell division. Start off with the treatment drug called chemotherapy drugs such as vincristine, which is also another name for oncovin, stops mitosis by disrupting the binding to tubulin and stopping the microtubule assembly. In eukaryote cells, the process of nuclear cell division is where the parent cell divides into two identical daughter cells. That is called mitosis. This process separates the duplicated genetic material and is done at the nucleus. The stages are in order: prophase, metaphase, anaphase, and telophase. There are protein structures called spindle fibers that are formed early in the cell division. There are also microtubules that are polymers made of tubulin protein. They originate from the centrioles that are structures located in the centromere of the chromosome. The mitotic spindle's main function is to be the sort of framework and attachment to the chromosome. The mitotic spindle is disrupted by the chemotherapeutic drugs such as vincristine. They are made of microtubules that are polymers of tubulin. When the mitotic spindle is disturbed, the process of mitosis becomes uneven. Irregular attachment, aligning, and assortment of the chromosomes. The sorting and segregation of the chromosomes are affected. Cell's arrest takes place in conditions that lead to cell death. This unorganization increases the occurrence of aneuploidy, or incomplete sets of chromosomes in daughter cells. Aneuploidy is the major characteristic of cancer. Aneuploidy is presence of an abnormal number of chromosomes in a cell. Polyploidy is when a cell has more than two paired sets of chromosomes. Tubulin is a type of protein which polymerizes into long chains of filaments. Oncovin is also named vincristine, which I stated earlier. It is a natural alkaloid that is isolated from catharanthine. The mechanism of oncovin is an inhibitor of the microtubule dynamics. No, it's an inhibitor. What was it? Inhibitor of the microtubule dynamics that will cause a mitotic arrest and eventually lead to cell death. Oncovin tends to bind to the B-tubulin subunits at the positive end of the microtubule. At this region, which is often called the Vinca binding domain, this Vinca binding domain is a domain that is binding in the microtubule, which decreases the dynamics of the microtubule and its overall composition. Therefore, it is often assumed that the proper assembly of the mitotic spindle, which could be prevented, and the tension that is in that mitotic spindle at the connective cores of the chromosome could be reduced. This type of binding, the unwanted cell division, can be controlled, so it is used in cancer cell treatment, which is an example of why oncovin is used, or aka called, which is also called oncovin and vincristine. And on my slide, I'm about to present the 3D structure. As you can see, I'm about to zoom in. And there goes the 3D structure of vincristin or oncovin.